Good afternoon, ladies and gentlemen. My name is Tianlu Zheng. Our team comes from Kaohsiung Medical University. Today, we will show an interesting topic. The title is Universal Antibody Lock Enhance the Disease Selectivity and Safety of Antibody Drug. We know the antibody drug has become the major trend of drug. The global top 10 drug in 2020 has five antibody drug. The antibody drug neutralize self-antigen for the treatment of disease, neutralize TNF-alpha, anti-PD-1, and VEGF. However, long-term use the antibody drug cause many side effects. Okay. Long-term systemic neutralizing antigen will cause side effects because the half-life of antibody has 20 days. When we systemic inject the antibody into our body, okay, the antibody will deplete all antigens function and cause some side effect. Example one is approved drugs has a serious effect, like Remicade and Humira, anti-TNF-alpha antibody to neutralize TNF-alpha to treat rheumatoid arthritis (RA) disease. However, systemic depleted TNF-alpha will induce hepatitis B/C reactivation and increase the risk of malignancy and serious infection, like TB, bacteria, and virus infection, to cause 36% patient infection and 17% patient need to stop the treatment. The second example is a cause clinical trial failure, like immune checkpoint stimulator, the 4-1BB antibody. The antibody can activate T cell. However, the antibody clinical trial has stopped because serious liver toxicity. Another way is to reduce dosage, but reduced dosage decrease the therapeutic efficacy of antibody. In fact, 123 antibody failed in clinical trial because serious side effects. So it's important to improve the selectivity of antibody drug. Our strategy, developing antibody lock to improve the selectivity of antibody drug. We autograft the hinge part, phone 第三位棒. We autograft hinge to antibody, to the N terminal of antibody, and use the protease peptide as linker to connect the antibody lock with antibody to form the pro-antibody. The antibody is inactive in vivo. Okay, when the antibody go into disease site, the protease will remove the antibody locker and activate pro-antibody to neutralize antigen in disease site. The strategy has three advantages. One is selective activation. Second is reduce the side effect. The third is increase the dosage of antibody. Okay, and then we also compare the hinge lock with binding peptide, our competitor. Okay, KMU use antibody hinge as a masking domain. We just autograft hinge to N terminal. Our competitor, CytoMax, use binding peptide as antibody lock The binding peptide screen from bacteriophage. Okay, and the binding peptide can binding with antibody as a lock. In the immunogenicity, because our hinge is autologous, we just autograft, the immunogenicity is low. And the binding peptide is an exogenous peptide and screen from bacterial display. The immunogenicity is high. And in a completely removed, our hinge antibody is a spatial adherence-based antibody lock. And the lock can be completely removed after protease cleavage. But the affinity adherence-based peptide has a high risk interaction with antibody. And for the widely application, the spatial adherence-based antibody lock can lock any antibody. But the binding peptide need rescreen from bacterial display library. And for the developing time of pro-antibody, our lock may be 2 to 3 months, but binding peptide maybe need more than 12 months. So the hinge lock is better than binding peptide, Cytomax. We also prove concept our strategy in rheumatoid arthritis model, RA model. We evaluation of therapeutic efficacy and side effect of Remicade and LAC-Remicade. Remicade is an anti-TNF antibody in vivo. Mice treat with PBS Remicade and LAC-Remicade and monitor the therapeutic efficacy. From the figure, we can see the blue line and red line have a similar effect. So the conclusion is that Remicade and LAC-Remicade has a similar therapeutic efficacy. And antibody LAC can reduce the risk of listeria infection. And mice treated with PBS, Remicade and LAC-Remicade, and then inject listeria bacteria. The results show the Remicade group mouse die during 5 days. But the LAC-Remicade is the red line. Mouse still have 71% survival. So the conclusion is LAC TNF alpha, it's LAC-Remicade, increased survival and equal efficacy. And similar results also shown in anti-TNF alpha, also shown in CTLA4, PD1, 4-1BB antibody. And the hinge patent has applied in 18 countries and getted in 15 countries. And we also developed some pro-antibody pipeline. Okay, from the table, the LAC-TNF alpha, LAC-PD1, and LAC-CTLA4, and LAC-CD52, these 4 antibodies has proved in vivo therapeutic efficacy and in vivo side effect. Okay, and the LAC-EGFR ADC look for partner. And GNS201 and GNS202, we have cooperation with Taiwan company. And the strategy has 3 applications. Okay, one is for the clinical trial stop or withdraw antibody, like Reactiva or anti-CD28. Our LAC can improve the side effect, okay, to produce a second generation of antibody drug, remarketing this kind of antibody. And second, for patent expired antibody like Remicade, Herceptin, our luck can revoke a new patent. The third, for some new drug but has a serious side effect, our luck can increase value, break through the market with high safety. And our strategy also have two business model can provide. One is we can license biobetter for company. Our bank, antibody bank, has more than 10 antibody clone like antibodies proof of concept. We can license out to get royalty. The second model is a co-development with company. Okay, a development for customers' antibody. We can do drug design, in vitro function, and in vivo function. And cooperation with some partner like CMO, CRO, development cell line for preclinical test, and then license out to get the royalty. Okay, and we have a we have a start-up team. We have a new company. Our company name is Precisement Antibody Biotechnology. It's a R&D biopharmaceutical company. We will be found in 2021, based in Kaohsiung, Taiwan, and focused on developing the safest and the most effective antibody therapy. I'm the chief officer, and Yun Qi Li is a CEO. She is a co-inventor for the antibody lock. Yan Zheng as a CTO, and Zhao Yan Wang as a medical advisor, and Catherine Zhou as a business and development advisor. And finally, our conclusion is we can provide good luck to make your antibody safer and give you luck. Thanks for your attention.